Yeah, so today is the 14th May 2020, and it's my pleasure to welcome Associate Professor Benedetta Bussolati from University of Torino, Italy. Benedetta is also the founder and current president of the Italian Society for Extracellular Vesicles, or EVIta. She has extensive experience in studies of stem cell biology and regenerative medicine, and includes the characterization of various stem cell types and their potential use. So today she will talk about her current work on the role of stem cell derived extracellular vesicles in regenerative medicine as therapeutic tools.
Okay, thank you. So good morning or good afternoon, everyone. And I really would like to thank Carolina for this initiative, which I enjoyed really a lot. And I think I can list it on one of the good things regarding this quarantine. So today I will talk to you about sources and mechanisms of EVs for the repair of the kidney, and I will try to drive you on a journey going from stem cells to urine. So my first question is, do stem cells EVs and urine EVs look alike? And I'm sure your answer would be not that much, not more than Schwarzenegger and EVIta look alike. But I would try to convince you that they indeed may have some common properties. Okay, so today we will be talking about kidney, as I'm a nephrologist, and we were mainly involved in looking for therapy for kidney pathologies, which include acute kidney injury, or AKI, which is a sudden and abrupt loss of the renal function due to many different causes, including also infectious diseases as COVID. And we will be talking about chronic kidney disease, which is progressive and irreversible pathology leading to the complete loss of renal function, going to dialysis or transplantation need. So mesenchymal stem cells were studied and appearing a good therapeutic option in our lab. But we quite soon understood that the effect of the injected cells was not dependent on stem cell tissue localization. And you can see here in this image the labeled cells going even to the kidney, but not being integrated into the functional structures, so remaining in the interstitium, so aside. And so, to make a long story short, we arrived to the conclusion that this effect was mainly mediated by soluble factors, and I mean, we were not the only, whole community was conveying to that, and that EVs from stem progenitor cells could really represent a unique tool to promote organ regeneration, as they could recapitulate the effect of the stem cells on the target tissue, and they could display a long-term reprogramming that was mainly mediated to the transfer of their content, and in particular of xRNAs. And I like to cite two seminal papers, one from the group of Rajajak, who was really the first studying the EVs, that at that time we were mainly microvesicles from embryonic stem cells, and then this second paper in which I was also involved from Camusi group from EVs from endothelial progenitor cells. So this is an image from the first paper by Stefania Bruno showing the effect of EVs in renal regeneration in a model of acute kidney injury. And so this is a rapid model in which the animals get damage at day 3, and at that moment we injected the cells and the EVs. And I would like to point out that in those experiments, we were trying to set up a dose, and we choose to inject a cell equivalent dose, meaning that we injected the number of EVs that were produced by a therapeutic dose of cells overnight. And it was clear that we had a very beneficial effect of EVs in accelerating the renal function, as you can see here, the decline of retention parameters that mean the gain of renal function. And we also were studying the localization of EV after injection into the tail vein. And so this is a healthy animal, and as you can see here, the injected and labeled EVs do not localize much into the kidney, and you can confirm that also at the autopsy. They mainly localize in the liver and in the spleen of the animals. But at variance, when we injected in the acute kidney injury animals, the vesicles localized into the renal tissue, and this is also confirmed by looking after sacrifice to the kidney that you can see here, and by immunofluorescence, the vesicle look really entrapped into the renal tubule cells, which are the damaged one, as soon as 15 minutes after the injection. So a number of studies have been done since then. Here you can see a list of the majority of them. And although it's not easy to compare those studies, as they are mainly using different types of evaluation of the dose of the V and different types of administration, the results are mainly confirmed. So yeah, that's an important point. You can see some people use micrograms to check for the EV amount. Other people use the number of EVs, and that's what we do in our lab. And the injection was single, were multiple, were intravenously, or also into the renal capsule. But the majority of studies were concordant to show that EVs could have a regenerative effect, promoting the tubular cell proliferation, the differentiation, acting also on endothelial cells, promoting angiogenesis, and limiting the recruitment of inflammatory cells, the fibrosis, and apoptosis and stress of the cells. So all these studies set up the stage for a first clinical trial that was performed. This is an Egyptian organization in diabetes-induced chronic kidney disease, in which 2 groups of 20 patients were treated with 2 different doses of MC-derived EVs interarterially. And for 1 year, they followed the GFR, which means the glomerular filtration rate is the function of the kidney. And in the control group, they observed a stationary or even a decline of the function, whereas in the treatment group, they observed amelioration, at least for some points of the renal function. So this is obviously to be confirmed. More trials are needed, more controlled trials are needed, but it's an interesting aspect. So I will just talk briefly about the possible mechanism of action of these stem cell derived EVs. So we were mainly interested in the role of microRNAs, and to show the proof that they were involved, we generated DROSHA knockout mesenchymal stem cells that have an impairment in the maturation of microRNAs, as DROSHA is pivotal in the maturation from pre-microRNAs to pre-microRNAs. So you can see here the list of microRNAs that were downregulated in the EVs coming from the DROSHA knockout cells in respect to the control EVs. So Federica Collino in Camus's lab performed the experiments of treating Acute mice with mesenchymal stem cell EVs and DROSHA knockout EVs. And as you can see here, I'm not sure you're very familiar with these images, but the damage, the alteration, the profound alteration which is observed in the renal tissue of Acute mice was completely restored. This is a tissue showing no sign of toxicity, of damage in mice treated with mesenchymal stem cell EVs, and at variance, this was not gained in DROSHA knockout EVs, as confirmed by the quantification of the casts, of the necrosis, of the tubular necrosis, and a functional parameter, which is the bone, which was completely ameliorated with mesenchymal stem cell EVs and not with DROSHA knockout EVs. But I think the main message of that paper was related to the analysis of the transcriptome of the renal tissue. So we can see from this graph that the transcriptome of the tissue of mice treated with control EV was correlating with that of the healthy mice, as well as with that of the mesenchymal stem cell treated animals. And at variance, the transcriptome of the EV DROSHA treated mice was not different at all, was correlating with that of the acute kidney injury animals. So this data really tells us a profound effect of the extracellular vesicles at multiple levels on the ability of the tissue to undergo a profound regeneration. And I think this is really relevant. This is relevant especially in conditions of fibrosis and chronic injury, such as in chronic kidney disease, in which we observe a profound real alteration and change at multiple levels of the whole transcriptome of the patient. And this is just the analysis of the alterations, mainly due to methylations of a renal tissue in chronic kidney disease. So more recently, Cristina Grange wanted to test also in a chronic condition the effect of EVs. And so she set up a model of diabetes, and in this case, she set up a curative model, meaning that mice were treated with streptozotocin to induce diabetes, as you can see here, and then diabetes was induced and set for 1 month before starting the injection of EVs. That was a multiple injection for 4 weeks, and the animals were sacrificed at 60 days. So the animals at that point, at the sacrifice, had a profound change in the expression of some pro-fibrotic genes, such as alpha-aspartate, EVIta, and collagen, which was reversed by the administration of the stem cell derived EVs, particularly of the mesenchymal stem cell EVs. Here you see collagen and TGF-beta. But going deeper in the analysis, we can again see that different sets of genes that were changed by the diabetic condition, as here upregulated or downregulated, were really reverted to the normal condition, to the control condition, by the EV administration. And going into detail, some of them were obviously linked to the induction of fibrosis, such as modulation of collagen, of metalloproteinases, serpin, and many others. So what we can say from all these experiments regarding the renal tissue is that in pathologic condition, the injection of EVs from stem cells into the circulation of the animals can represent a therapeutic tool. The kidney is profoundly modified and regenerated by those vesicles that reach the tissue possibly by an increasing permeability of the peritubular capillaries, as we cannot see, we cannot imagine a passage to the glomerular permeability barrier, at least if the glomeruli are not completely damaged. So this also implies the fact that in normal condition, we have a closed compartment in the nephron in which the vesicles that are inside the urine, the capillary lumen, mainly derive from the cells of the kidney. And so we in the meantime, also got interested in these aspects of the EVs that are present in the urinary lumen. And there are several studies that start also to show that these vesicles, that obviously are very important as a marker for the condition of the kidney, can also have some role in transferring information along the nephron districts. And in particular, some studies were showing that EVs are involved in the communication transferring anti-inflammatory phenotype from one tubular aspect to others, and they also may transfer functional transporters toward the tubules. And also there are some papers showing that if we collect the cells from the urine and then we culture and we prepare vesicles, they might have some regenerative properties. And just to briefly tell you that vesicles along the different tubular segments express different markers, which are mainly related to the type of cell which is producing them, to understand the fact that we started working with urinary EVs, is isolating them and characterizing them. And so we're isolating vesicle from urine of normal subject by ultracentrifugation, and also by a subsequent floating, as we wanted to get rid of possible contaminants in the urine. We have thermophosphate protein, which might be entrapped also during ultracentrifugation. Those vesicles, as I was telling you, express some markers of the different segments of the renal tubules, underline the fact that they mainly derive from the cells of the tubules, although some of them might also derive from the bladder and from the prostate, as Carolina and I were also discussing before. But you can see here the expression of markers of the tubules, of the glomerulus. CD24 is also quite expressed by extracellular vesicles in the urine, but no marker of vesicles from the serum is present, such as CD45, vesicle from leukocytes, or vesicle from platelets expressing CD42B, or vesicle from the endothelial cells. So these results were obtained with beads, so are not quantitative, but just semi-quantitative results that were also confirmed by Western blot. And so at that point, we decided to check whether the injection of vesicles coming from the urine could also have some effects in the mice undergoing acute kidney injury. So we again used a very simple model, a very quick model of acute kidney injury. The mice underwent damage at day 0. The peak of damage was at day 1 when we injected EVs, 1 per 10 to the 8, and at day 3 we sacrificed the animals, and we injected 4 different types of EVs. EVs from ultracentrifuge urine, EVs from urine that underwent ultracentrifugation followed by floating, EVs from mesenchymal stem cells obtained by ultracentrifugation, and EVs from fibroblasts that were our negative control. Just to check the fact that EVs could reach the kidney, we performed some imaging studies, and again we confirmed the fact that in the healthy animal, once injected into the circulation, EVs could not get to the kidney, but at variance in the acute kidney injury damage mice, we had a huge localization of the EVs after 24 hours. Okay, so these are the results. You can see again the increase in creatinine and bone retention marker in the damaged animal treated only with vehicle. And then you can observe that not only mesenchymal stem cell EVs, but also urine EVs, both from ultracentrifugation and from floating, were performing a very good therapeutic effect. And fibroblast EVs were not significantly different from vehicle, and this is also confirmed with another parameter, which is Boone. And also looking at the histology of the kidney, it is clear that we have a profound damage with necrosis and appearance of these casts, which are protein aggregates within tubules that impair filtration in the vehicle, in the fibroblast EVs, but these are absent in the animals treated with mesenchymal or urine EVs. We also checked the effect on tubular cell proliferation, which is an important parameter for the regeneration of the tubules. And again, we had a great increase in proliferation due to the administration of EVs from urine and from mesenchymal stem cells. And in the meantime, also the parameters, classical parameters of renal damage were decreased by the injection of the UEVs and mesenchymal stem cell EVs. You can see here in GAL, PI, SOX9, decreasing caspase, and in several inflammatory cytokines and NFKB. So what could be possibly the mechanism of action? So obviously several mechanisms could be possibly involved, but we got quite interested in a factor which is kidney specific. And this factor is called Klotho and is an anti-aging factor mainly produced by the kidney. You can see here mice that do not express Klotho in the kidneys do not have Klotho also in the circulation, because Klotho is produced, as we are going to see, also a soluble factor. So going a bit deeper into Klotho, the name of Klotho is under the name of a Greek mythological figure which is involved in spinning the thread of life. Why that? Because Klotho is really an anti-aging factor. It's really controlling our aging. So mice that were knocked down for Klotho were going under several alteration, endothelial dysfunction, vascular calcification, atherosclerosis, and their lifespan was reduced up to 15 weeks in respect to normal 2.53 years. Klotho, as I was saying, acts both as a transmembrane receptor and as soluble receptor, acting in this way in a hormonal fashion. So it can be a co-receptor for FGF23, and as cleaved, it might present in a longer form or in a shorter form, and could be also directly secreted as soluble form. And it's still really unknown, the mechanism of action. There are few papers going into the dissection of the mechanism of action of Klotho, but for sure it's quite relevant fact that it might inhibit fibrosis and oxidative stress. So the vesicles of the urine contain Klotho. So they contain some of the full-length Klotho, but they may contain the short soluble form of Klotho. And I'm very proud to also show you the face of the urinary extracellular vesicle co-expressing Klotho and CD81, also because this was the last experiment we could do before the lockdown in Italy, using the super resolution microscope from ONI. And so you can see here two vesicles that show expression of Klotho in blue and CD81 in red. Sorry, the scale bar went not very clear. This is 50 nanometers. And one vesicle that was not positive for CD81 and only positive for Klotho. So the expression of Klotho is quite strong in the urinary extracellular vesicle. So in kidney pathology, the decrease of Klotho could be really an impact for kidney regeneration in the acute damage, for fibrosis and the chronic damage, as well as obviously for the whole individual losing Klotho production. So we first checked whether the administration of urinary EV could ameliorate the expression of Klotho in acute mice. This is the expression in healthy animals. This is the loss of the expression in acute kidney injury. And it is clear that administering urinary extracellular vesicle, the expression of Klotho was really regained, far more than by administration of the mesenchymal stem cells EVs could, that showed some reappearance of Klotho, but not that much. And this is also confirmed by analysis at the messenger RNA level. After 4 hours, we can clearly see that Klotho starts to regain in animals treated with urinary EVs, and also the protein level, the tissue was expressing Klotho significantly higher than in animals treated only with the vesicle, which was obviously maintained also at 48 hours, as we can see here. So to prove the real role of Klotho in the urinary extracellular vesicles, we took advantage of a collaboration with Paul Robbins that provided us with the urine of mice that were Klotho knocked down. So we injected in our model normal extracellular vesicles from mice, knocked down extracellular vesicle for Klotho, and those knocked down extracellular vesicles plus soluble Klotho. And the experiments really confirmed the fact that Klotho was relevant, as the murine extracellular vesicles showed regenerative effects. As you can see here, decline in the retention parameters, in the number of cast and necrotic tubules, parameters of damage, and this was not reached by the Klotho knockdown murine extracellular vesicles, which did not show any sign of regenerative effect. But in the animals treated with murine extracellular vesicles plus soluble Klotho, we saw a similar effect to the normal urinary extracellular vesicles. Okay, we wanted to also compare the effect of Klotho present in the urinary extracellular vesicles with that of soluble Klotho. This was actually a request by the reviewer, but was also a strong rationale, as there are many papers showing that injection of recombinant Klotho could help the regeneration of the kidney, but the quantities that are reported in those articles usually are in the range of the micrograms. And so we decided to use soluble Klotho in the range that was present in our extracellular vesicles. So we compared 1 picogram of Klotho, injecting the amount of vesicles that were containing that amount. We also compared the effect with that of Klotho contained in vesicles derived from cultured tubular epithelial cells that were indeed containing some Klotho, and soluble recombinant Klotho. Okay, and again, we had a very nice effect with the urinary extracellular vesicles, with the proximal tubule derived extracellular vesicles, but the effect with the recombinant Klotho at that quantity was not any different from that of the untreated animals. These are again all the parameters of retention and parameters of histological damage. So I think we were quite convinced that Klotho present in the vesicle really makes a great impact in the effect of those vesicles. And so the last aspect that we thought could be of interest is the possibility to engineer vesicles with recombinant Klotho. And so we choose the fibroblast, as in the previous experiments. You remember, fibroblasts were not effective, and we added Klotho to those fibroblasts. So we engineered EVs with Klotho. Here is the Western blot showing the presence of Klotho within the vesicles, both the short and the long form, and we injected the AKI animals. So the first important parameters is the expression of Klotho by the kidney, and here you can see that Klotho was regained, immunohistochemistry, and also at Western blot analysis. So renal cells regenerated could express Klotho, and also by functional parameters, fibroblast EV that were engineered with Klotho had a very good effect in promoting regeneration, in reducing markers of damage, and histological markers of tissue necrosis, and finally promoting the proliferation. Okay, so I would like also to show this this slide from the previous WebEVTalk from Oscar that really was helpful in providing some insights into the possible effect of Klotho engineered on the vesicle. Obviously, the Klotho expressing vesicles could work by a better delivery of the factor to the tissue, by increasing its half time. But I think it's also very interesting to analyze the aspect of a possible signaling platform of the Klotho present on the vesicle on the target cells in respect to the soluble factor. So this will be for sure one next point to evaluate, and I really thank Oscar and the EVITalks for providing new ideas. Okay, so this is just the summary of our studies, in which we show that EV isolated from urine had an effect on renal regeneration, increasing the expression of Klotho, decreasing inflammation, promoting proliferation, and the same could be also obtained engineering EVs with the soluble Klotho. In conclusion, this could also have some other possible therapeutic effects and horizons, engineering autologous EV with Klotho in a different subset of pathologies, as Klotho it's involved in aging, cognition, cancer, diabetes. And anyone who is interested in collaborating, please contact me. So just to acknowledge a lot of people which have been involved in those studies, for sure Christina and Ellie and Veronica, who was not with us in that evening for the Klotho extracellular vesicle experiments, all the study in Professor Camusi's lab, also Licia Brizzi, who is responsible for the diabetic study that I showed you, collaboration with Paul Robbins, some funding agencies, including the European Union, Regione Piemonte, Unisite, and I'd just like to acknowledge this wonderful society, EVIta, for the intense discussion and the opportunities. And you all for your attention, and I hope you will have questions for me.
Thank you so much, Benedetta. Thank you very much for this interesting presentation. So this talk is now open for discussion. We do already have some questions. This is on the chat box there. So please put your question in there. I will let you to ask the question verbally as well. So Kalyani Patil, could you unmute yourself and ask your question, please? If not, I'll ask the question. Do EVs play a role in hormonal deregulation in acute kidney injury and restore normal hormone function, I guess, from the kidney? So could you comment on that, Benedetta?
I think, I mean, it's difficult to check in our very short time experiments hormonal dysregulation. I mean, obviously erythropoietin and vitamin D could be altered, but we never checked that in our animals. That's probably in 3 days or 7 days, whatever. But in chronic condition, it would be of great interest. I think no one ever checked that. It's a very good suggestion.
So we have another question, Genevieve Bart. Can you unmute, Carolina? Allow people to unmute. Yeah, there you go.
Sorry.
Good.
Can you hear me?
Yes, Genevieve,
Yes.
we can.
Yeah, Genevieve Bart from University of Oulu. I'm sorry about the cat. I can show my face if you want. I was curious to know, how can you control the amount of Klotho in your vesicles? Do you expect it to be like evenly distributed, or is it just random?
So for the moment, what we checked was the amount of Klotho in pools of urine. We never inject a single subject. We always pool the urine, and we found obviously variations in the different pools, but I mean, every time we were checking the amount, and we know it was ranging from 1 to 10 picograms per 10 to the 8 vesicle. So if that's what you mean, if you want to know single vesicle, then that's a different story. We are starting to check also the co-expression of the vesicles with other renal markers, because we would like really to understand which type of cells produce Klotho. And I think with super resolution microscope, this could be checked.
So that's interesting. Is there also a subpopulation of vesicles that express Klotho and other vesicles do not?
Yes, yes. So not all the vesicles in the urine are positive for Klotho.
So just one more question. Are you expecting this these Klotho-positive vesicles to be coming from stem cells that are in urine, for instance, or is it completely a stupid question?
No, I think it's a relevant question. Actually, we don't know, but we know that Klotho is released from cells of the proximal tubules and also some distal tubule cells. But we don't know which one. It could well be that those cells are the progenitors and not normal cells. So that's something, I mean, quite open as a question.
Thank you. So we have another question from Lin Li about administration. Lin Li, are you there?
Yes.
Yes.
This is Lin Li from South University, China. I'm happy to be here. I'm just wondering, what's your opinion about the EV administration approach in therapy of kidney disease? I mean, how to promote the kidney localization by peritoneal injection or tailway any difference by these two approach. And also extracellular vesicle is thought to be difficult to filter through the glomerular filtration barrier. Do you think this will limit the usage of extracellular vesicle in therapy of kidney disease?
Okay, so regarding the route of administration in the mice, we checked and we compared intraperitoneal with the tail vein, and tail vein had a superiority. That's why we chose that, even though vesicles circulate also after the intraperitoneal administration. And so regarding the limit of extracellular vesicle to pass the glomerular filtration barrier, obviously in normal condition they cannot pass. And even in chronic kidney disease, as you say, that could be a problem as glomeruli become fibrotic. So it's even more difficult. We can imagine a passage in acute situation of glomerular disruption, but we have the peritubular capillaries. So I really believe that possibly vesicles could reach the tubular cells on the other way, passing by the peritubular capillaries. The delivery to the glomerular cells, that's I mean, that's something that it's not really known. There could be some passage from the endothelium to the podocytes that just stick on the other side of the membrane. Maybe some mechanism of transcytosis, so we don't know. That's another important point that would be quite relevant for the therapy in humans. Sure.
Thank you. So I think regarding biodistribution, we talked about this before in these sessions, and if you inject IP, you get a very strong administration to the lymphatic systems, including thoracic lymph nodes and local, whereas if you inject intravenously, you have access to the endothelial layer. And that follows up to the next question, which is Anna Paulina, who is asking a question as well. You can unmute yourself. I can unmute you, and you can speak, Anna.
Okay, thank you. Thank you, Benedetta. I was just wondering, why is it that the EVs goes to the injured side and not to the spleen like in the healthy model?
So it could well be just a matter of permeability, but we also like to imagine, and there are studies providing some evidence that this could be also due to the expression by the damaged tissue of receptors, as also stem cells do, that bind the vesicles, so the vesicles might be more prone to enter into the damaged cells and to stay into the damaged tissues.
Okay.
Thank you. I think when you again look at biodistribution to injury, it doesn't matter which injury there is. If you have a brain injury or other tissues that are injured, the vesicles do increase their homing to that tissue if there is injury there, and that's sort of a known fact. Tim Williams, please unmute yourself and ask your question.
Hi, thanks very much. It was just a general question, really, about the experimental design. Have you ever tried to deplete your extracellular vesicle preparations of EVs and then use that as another control in order to sort of look at how important the actual EV itself is in these experiments?
We never did it in vivo. We did it in vitro in some experiments. Just checking the supernatant, so not really depleting, but just checking supernatant. So that was our control. Maybe you have suggestions how to deplete EV preparation?
Well, I mean, it's something we've talked about in it's something we've talked about in our lab before, and in the past we've done work where we've sonicated the preparations to try and disrupt the EVs, and we've talked about whether you could use some kind of beads to pull down the EVs. But the problem with that approach, I think, is that probably different EVs have different express different markers, and so you may not deplete all of the types of EVs. So I was just wondering if you ever tried it, and if you had, what your strategy was.
No, not really in that way. Yeah.
So I think this is a very, very important question that I think the whole field is struggling with a little bit. If you look at the contribution of the vesicles in the isolate from cells supernatant, and think about what else is there, right? Non-vesicular, and the subpopulation of vesicles. What is it that really contributes to the biology? I can say that in our lab, we have really purified the membranes of vesicles and could retain some biology with just keeping the membranes without actually microRNAs or any other, or at least reduced cytoplasm or vesiculoplasm proteins inside of the vesicles. So, there's a lot to do to understand that, Tim. It's a good question. Benedetta, you want to comment?
Yeah, you're right. I mean, the membranes for sure increasing number of papers showing activity, and also possibly the content in a soluble form. So, it's difficult to dissect the role of the various components of the EVs. That's something that it's relevant, although possibly the EV itself has advantages in a various ways in respect to the single components of it.
So when you look for a single protein, and biology is single protein released by cells, it makes some kind of sense. But for vesicles, there's a whole array of molecules that contribute to the biology, and it becomes a little bit difficult to dissect those.
And then I'm sure vesicles from a biological liquid also are sticky and transport, we know, microRNAs or proteins. So that's something that, it's a physiological mechanism. It's not really contamination because it happens in vivo, in the urine or in the plasma. So it's probably also relevant for the activity.
Mohammed Nawaz has a question. Please unmute yourself. It's one of the favorite topics of the whole scientific community right now. Not surprised. Mohammed?
Mohammed
I know Mohammed
seems to have
sometimes had
issue with the microphone.
Are you there, Mohammed?
He has some issue with the microphone, yeah.
Okay. So if you can, so how about kidney responses to SARS-CoV-2 infection and COVID disease? Could you comment on that? I've heard from intensive care doctors that they do see kidney failure sometimes.
Yeah, so coronavirus affects heavily the kidney. It induces a large variety of different pathologies, including glomerular damage. It affects, the ACE2 receptor is present in the podocytes, so it may induce a sclerosis of the glomeruli. It might also induce an acute kidney injury, so a complete loss of the kidney function, which can be acute or can possibly also evolve toward a chronic one. So it really has a big impact, much more than it was expected at the beginning.
So Carolina, you have a question as well?
Yeah, thanks. Maybe I'm missing some information, but with the urinary EVs, since it seems to have a better therapeutic effect on the acute kidney injury from the MSC, and taken from that results, what do you have a comments on where the cellular origin is important, or is it Is it enough just have a certain like protein expressed on the EV like the Klotho for therapy?
Yeah, that's I mean that's not easy to comment for sure. I mean the urine, it's a very heterogeneous pool of EVs from a lot of sources. So I'm not sure this would be ideal for therapy. There's problem of infection, there's problem of heterogeneity. So we don't know, but for sure that, I mean, the identification of a renal marker, which is produced by the kidney and has an effect on the kidney itself. So I think it's the beauty of Klotho is the fact that it's a renal specific protein. And the fact that the kidney has a lot of receptors for Klotho might be relevant. So the idea of engineering EVs with a recombinant protein might be more likely for therapy than using the urine, possibly. But it's also important to understand mechanism of cell-to-cell communication in the kidney.
So we have another question from Sharad Kolia. Please unmute yourself and ask your question.
Good morning, a brilliant talk. My question is that would the source of urine actually have an effect on the therapeutic effects observed? So for example, in terms of gender, diet, etc., on the EVs that you isolate from the urine?
You can ask a question in another way. So are there huge differences in the EV structure from young people, old people, female, male,
yes,
well-trained, untrained, so on and so forth? So Benedetta, tough question, right? But it's important.
Yeah, I have to admit that the origin of the urine was mainly from people in the lab, so from young, healthy subjects. Okay. So possibly old subjects. We already know that all subjects have different markers. There are some papers showing differences between female and male, but not in our hands, not on Klotho expression, but for sure old and young we have differences. Yeah.
Thank you.
I have an additional question here. Could Klotho vesicles be helpful for other organs?
Yeah, that's something that I think is of great interest. For aging in general, we would like to check that because it has a great impact on aging, on cardiovascular system, and as well as on the cerebral nervous system, central nervous system. So dementia and those pathologies.
So that was the last question that I can see listed. Any additional question, just ping us on the chat. I'll give you 5 seconds. If not, thank you so much, Benedetta. It was a pleasure, and thank you everyone for attending. It's always great to join these. They're a little bit of sunshine in the dark days that we are going through right now. And everybody, stay safe, okay? And we'll see you soon. Bye. Bye.